AlphaFold has revolutionized structural biology in just 5 years, solving one of science's grand challenges. At its core, AlphaFold solves protein folding, predicting how a chain of amino acids transforms into a precise three-dimensional structure. For half a century, predicting protein structure from amino acid sequence remained unsolved. A protein starts as a linear chain of amino acids, encoded by DNA. But how does this chain know how to fold? The chain spontaneously folds into a specific three-dimensional structure, determined by physics and chemistry. This is biology's central dogma extended. Sequence determines structure, and structure determines function. Why did this problem remain unsolved for 50 years? Let's look at the numbers. A typical protein of 100 amino acids has roughly 10 to the 300 possible conformations. That's more than atoms in the universe. Multiple competing physical forces shape folding. Energy landscapes, hydrophobic effects, local and long-range interactions all matter. Before AlphaFold, scientists tried several computational approaches with limited success. Traditional methods like homology modeling, threading, and ab initio prediction achieved only 40 to 60% accuracy at best. X-ray crystallography and cryo-electron microscopy remain necessary, but they're slow and expensive. Each experimental structure determination takes months of work and costs tens of thousands of dollars. In 2020, AlphaFold 2 achieved a stunning breakthrough at the CASP14 competition. AlphaFold 2 achieved 90% accuracy on CASP14 targets, far surpassing all previous methods in a single leap. What made this breakthrough possible? The answer lies in the architecture. AlphaFold 2 combines evolutionary information, pairwise attention mechanisms, direct 3D coordinate prediction, and iterative refinement. The pipeline flows from multiple sequence alignments through the evoformer neural network to predicted 3D structures. The key insight. When two amino acids are in contact, they co-evolve together across species to maintain the interaction. In aligned sequences from different species, positions that vary together likely interact physically in the folded structure. DeepMind didn't stop at publishing the method. They predicted structures for over 200 million proteins. The AlphaFold database now contains over 200 million predicted protein structures, freely available to all researchers. This is a 1,000-fold increase over the roughly 200,000 experimental structures in the protein data bank. Over 2 million researchers worldwide now use the AlphaFold database, accelerating research across biology and medicine. Applications span drug discovery, understanding disease mechanisms, evolutionary biology, and biotechnology. In 2024, AlphaFold 3 extended the approach to predict interactions between proteins and other molecules. AlphaFold 3 can now model proteins interacting with DNA, RNA, and small molecules like drug candidates. This dramatically expands the scope from individual protein folding to molecular complexes. AlphaFold 3 uses a diffusion model architecture, similar to image generation models, but for molecular structures. The model learns to denoise random atomic coordinates into accurate molecular structures through iterative refinement. How do we measure if a predicted structure is correct? Several metrics are used. Root mean square deviation measures average distance between predicted and true atomic positions. RMSD measures how far predicted atoms deviate from their true positions. Less than 2 angstroms is considered excellent. GDT, or Global Distance Test, measures the fraction of residues within certain distance thresholds. AlphaFold predictions often match experimental structures to within experimental error around 1 to 2 angstroms RMSD. AlphaFold also predicts its own confidence for each residue, called PLDDT. Structures are colored by confidence. Blue means high confidence, yellow means medium, and red means low confidence. PLDDT ranges from 0 to 100, with values above 90 indicating very high confidence predictions. Confidence scores help researchers know which parts of a prediction to trust and which may need experimental validation. Despite its success, AlphaFold faces challenges, especially with intrinsically disordered regions. Many proteins have disordered regions that lack fixed structure and exist as dynamic ensembles. Can AlphaFold predict these disordered regions? Not really. It's trained to predict single structures. Intrinsic disorder means there is no single structure to predict, just a distribution of conformations. AlphaFold sometimes generates plausible-looking structures for disordered regions, called hallucinations. Low PLDDT scores flag these. This is why experimental validation and checking confidence scores remain essential. AlphaFold Multimer extends predictions to protein complexes with multiple chains. Many proteins function as complexes of multiple chains that must assemble correctly. Predicting the interface between protein chains is particularly challenging because it depends on subtle interactions. The interface between chains involves specific contacts that determine binding affinity and specificity. Let's estimate the computational cost of the AlphaFold database. 200 million proteins times roughly 5 minutes per prediction equals about 1 billion minutes of compute. That's about 2,000 GPU years if done sequentially, but massively parallelized across data centers. DeepMind used massive GPU clusters, processing thousands of proteins in parallel to complete the database. The energy cost alone for training and inference runs into millions of dollars. However, this investment saves billions of dollars in experimental structure determination that would otherwise be needed. Pushmeet Kohli, VP of AI for Science at DeepMind, sees AlphaFold as just the beginning. The vision is broader. AI systems that fold, dock, design, simulate, test, and discover across all of biology. The goal is AI as an active research partner that generates hypotheses and designs experiments, not just a passive tool. An AI research partner would generate hypotheses, design tests, analyze results, and iterate autonomously. A major frontier is moving beyond static structures to understand protein dynamics. Many proteins function by transitioning between multiple states, and understanding these dynamics is crucial. State 1 represents the initial state of the protein. State 2 represents the final state of the protein. In conclusion, AlphaFold has revolutionized the field of structural biology.